Hello everyone, my name is Carolina Garcia, and today I'm going to talk about insulin resistance and how it develops at the cellular level. I'll mainly be focusing on the PI3K ACT pathway and GLUT4 movement and how changes in these processes make it harder for cells to respond to insulin. My goal is to explain what normally happens and what goes wrong that it develops to type 2 diabetes in a way that feels clear even if you're not familiar with the subject. So moving on to our introduction. So the main point here is that in type 2 diabetes, the body still makes insulin, but the cells are not going to react to it. And because of that, glucose can't enter muscle and fat cells the way that it should. And this is why blood sugar levels are going to stay high. And meaning that over time, the pancreas is just going to produce more and more insulin. And what it is important here is that the problems in these pathways are going to start years before someone is diagnosed, which is why it's so important to understand them. So what actually happens inside the cell? And this is our first topic. The PI3K stands for phosphoinositide 3 kinase, and ACT is a protein kinase that helps control glucose uptake. But I'm going to call it PI3K ACT pathway from now on. So this pathway becomes weaker, and GLUT4 movement, the GLUT4 doesn't reach the membrane. So, and that's basically the core problem. So if you look at the figure on the right here, it shows the difference between normal cells and insulin resistant cells. So on the left, insulin helps glucose actually enter the cell, and on the right, even though insulin is going to still be there, the inside signaling is too weak, so glucose can't get in. Quant et al. 2018 showed this drop in pathway activation. So moving on to topic number 2, we're going to talk about normal PI3K ACT signaling. So here I'm showing how the PI3K ACT pathway works when everything is normal. Basically, when insulin binds to this receptor on the cell, it's going to activate these IRS proteins. And they're kind of like messengers that pass the signal inside the cell. So when that signal starts, PI3K jumps in and converts a lipid called PIP2 into PIP3. And that PIP3, it's important because it brings AKT2 to the membrane so that it can turn on. And once AKT2 turns on, it actually going to help the cell move GLUT4 to the surface. So if we take a look at the figure in the left here, you can see that whole flow happening from left to right. So insulin activates the receptor, IRS proteins get triggered, PI3K creates PIP3, and then ACT2 gets activated. After ACT2 turns on, it sends signals to all these different proteins in the boxes at the bottom. And that's what eventually leads to GLUT4 moving to the membrane so glucose can enter the cell. So ACT has different forms, but ACT2 is the main one that's going to control glucose uptake. So when ACT2 activates properly, GLUT4 vesicles are able to move from inside the cell to the membrane. So when AK2 activation is incomplete, the vesicles are going to stay inside. Van Gerven et al. 2023 showed that lower AK2 activity leads to weaker GLUT4 movement, even when insulin is present. So in insulin resistant cells, several steps are going to be weakened at the same time. So IRS proteins may not signal correctly, PI3K activation could drop, and PTEN removes PIP3 too fast. So without enough PIP3, ACT never fully activates. Take Naka et al. 2019, which is one of the sources that I use, it shows that this directly reduces glucose uptake in skeletal muscles. And Sarkesh et al. 2025 found that higher PTEN levels in adipose tissue are linked to pure insulin sensitivity. So if you look at the figure, we can actually see where this restructure starts to happen. So the red blocks and the arrows show exactly which steps fail. So up at the top, IRS signaling is weakened. So in the middle, PIP3K activity is lower and less PIP3 is made. And on the right, PTEN is removing PIP3 too quickly. Down at the bottom of the figure, you can see that ACT doesn't fully activate it, and it's going to affect the proteins that depend on it. And altogether, the figure shows how the pathway loses strength at multiple steps. So continuing with topic number 3, when ACT signaling starts to weaken, a lot of things downstreams are also going to be affected. So ACT normally activates proteins that help with metabolism and glucose uptake. So when that signal isn't strong, that means that those proteins don't turn on the way that they normally should. And one of the biggest changes is that fewer GLUT4 transporters are going to make it to the membrane. And if GLUT4 doesn't get to the surface, the cell basically loses its ability to pull that glucose in. So take Naga et al. 2019 shows this very clearly in muscle cells. So when I2 activation drops, glucose uptake drops a lot too. So even though insulin might still be there, the cell can't respond the right way anymore. Something that we have to keep in mind, and this is topic number 4, is that GLUT4 actually has to move through the cell to get to the membrane. It uses microtubules and actin almost like little pathways. But in insulin-resistant cells, that movement is going to be slowed down, right? A lot. Diaz-Vegas et al. 2023 used a tracking assay and found that GLUT4 vesicles just don't move the way they should. So even when some signaling is still happening. So the problem isn't just the signal, it's also the movement. So continuing with topic 4, we can see that in figure number 4 here on the right, it's showing what actually happens when RAP10 is reduced. And you may ask yourself, okay, what's RAP10? And RAP10 is basically a protein that's going to help move GLUT4 vesicles toward the surface of the cells. So in the first panel, you can see that RAP10 was reduced successfully, and that's really all that this part is really showing. The main part is the line graphs, and the black line is what normally happens. So when insulin is added, more GLUT4 reaches the membrane over time. However, when RAP10 is knocked down, the lines don't go up as much, so that means that GLUT4 can barely move. So the big idea here is that the cell still has GLUT4 inside, but without the RAP10 protein, it doesn't reach the surface, and this is why insulin doesn't work as well. So for topic number 5, there are some signals that can help this pathway, and there's some that can make it lower. So as we can see, BMP2 and BMP6 make insulin sensitivity better by helping with PI3K ACT signaling, the activation. And as well as magnesium also helps ACT work normally. But however, inflammation, especially things like TNF-alpha and low magnesium levels, both can make the pathway weaker. Schraver et al. 2017, Oost et al. 2012, and Wan et al. 2018, which are the sources that I use for this information, all of these show that these signals this different signals affect the strength of insulin signaling. For our topic number 6, I want to talk about how insulin resistance is not going to hit every tissue the same way. So for example, in muscle, the main problem is weaker ACT2 activation and slower GLUT4 movement, right? So since Muscle is where most of our glucose goes after we eat. Even a small change makes a big difference. Tech Naka et al. 2019 showed that how quickly muscle loses glucose uptake when ACT2 drops. However, on the other hand, in fat tissue, PTN levels and inflammations are more of the issue. And Starkage et al. 2025 found that those fat tissue changes actually spread and make insulin resistance worse in other tissues too. So just to wrap everything up, the main idea is that insulin resistance doesn't come from just one single problem. It's gonna develop when several parts of the PI3K and ACT pathway starts to weaken at the same time. So when the signaling steps like IRS, PI3K, and ACT2 doesn't work the right way, the cell doesn't react to insulin as strongly. And the signal just isn't getting through. Juan et al. 2018 showed how this weaker signaling lowers GLUT4 movement. And then on top of that, the movement of GLUT4 itself becomes even slower. Diaz-Vegas et al. 2023 showed that the vesicles don't travel through the cell the same way. So even if signaling is still happening, the GLUT4 doesn't make it to the surface fast enough. So when you put these two things together, weaker signaling and slower trafficking, the cell just can't take in glucose normally. And over time, that's what contributes to insulin resistance and eventually developing to type 2 diabetes. So understanding both parts, the signaling part and the movement part, helps explain why this condition develops and why it gets worse over time. And here are the studies that I used for everything that I talked about in this presentation. And that's all for today. Thank you all for listening.